Hi everyone, my name is Gabriela Leite. I'm from the MAST Program at Cedars-Sinai in LA, and I'm here to present this poster where we are presenting the results of rats gavaged with a human duodenal microbe called Rothia Mucilaginosa in associations with change in the ovarian physiology and expression of ovarian genes linked to corporeal lutea and follicular function. PCOS, recently renamed to PMOS, is characterized by increased levels of androgens in the circulation, followed by ovarian dysfunction and infertility. Increased evidence from the gut microbiome study suggests that specific microbial populations may contribute for this signature. And we previously demonstrated that Rothia Mucilaginosa, a bacteria found in the human duodenum, induced a PMOS-like phenotypes in rats, characterized specifically by disruption in the estrous cycles and in alterations in ovarian androgen signaling pathways. However, we know that the ovary is a highly heterogeneous organ with a complex spatial organization composed of multiple cell types and specialized structures that may respond differently to specific signals. So because of that, we decided to investigate whether Rothia Mucilaginosa altered normal ovarian physiology at the gene expression level and spatial resolution. So we gavage reproductive age wild-type females prog dolly rats daily with Rothia Mucilaginosa for 4 weeks in a row. After 4 weeks, half of the Rothia group and half of the controls were euthanized. The other half were followed for another extra 4 weeks, but we stopped gavaging during this period. At the end of each of these time points, we euthanize the animals and we collect the ovaries. These ovaries were utilized for spatial transcriptomics. As a first set of results, we observed that after the initial 4 weeks gavage period, 55% of the rats from the Rothia group showed altered estrous cycles with arrested diestrous phase, compared to only 30% in controls. And this pattern actually persists during the phase where we stop gavaging these animals. At the ovary spatial level, we observed several modification gene expressions, especially those that were assigned to the corporeal luteum structures. When analyzing gene expression at the spatial transcriptomics level, we observed that 15 distinct cell aggregates were identified in controls compared to only 8 in Rothia. And sample-based clustering analysis showed that ovarian cells in the Rothia group during the prolonged phase, or the phase where we stop gavaging daily with this microbe, overexpress several genes, including the AKR1C3, which encodes a crucial enzyme that biotransforms androstenedione to testosterone, the active form, and also overexpression of the RLN1 gene, which encodes the relaxin-1, a peptide hormone crucial for pregnancy and reproduction when compared to controls. Spatially guided analysis using high resolution and the CT assist images revealed that most beings that expressed these genes were in the corporeal lutea. When compared to controls, the ovarian cells in the Rothia group during the prolonged phase also show a downregulation of the ENHA gene, especially in the follicular granulosa cells. This gene specifically encodes a protein called inhibin A and B, which are potent regulators of FSH, which is associated with preventing premature follicle depletion. So in conclusion, daily gavage with duodenal bacteria Rothia Mucilaginosa induce altered estrous cycles, as we showed before. And Rothia Mucilaginosa is associated with altered expression of specific ovarian genes linked to corporeal lutea and follicular function. Rothia Mucilaginosa is also associated with increased conversion of weak androgens, such as androstenedione, to their active forms, such as testosterone, and downregulation of genes involved in the FSH feedback loop. So Rothia Mucilaginosa-driven PCOS-like phenotype could be associated with follicle over-recruitment and burnout, impacting ultimately fertility. Thank you so much for watching our video.